Hello, everyone, and welcome to Chapter 2. So, as you recall, in Chapter 1, we spoke a lot about it generally, just the general principles of viruses. We spoke a lot about, what the, how they look like, the structure, the capsomers specifically, and how the capsomers really give, kind of this beautiful symmetry. Either it's helical, or either it's basically this icosahedral, or they have actually this kind of weird kind of symmetry. Then we also spoke about, are they enveloped or not? And then we also, did a big dive into what the different types of genomes that there is and how we classify them into different Baltimore kind of classifications. And we started to kind of, let's say, connect certain themes together of like, the structure. And then we also started to look about, okay, how the structure and then how basically the enveloped viruses can really give us a bit more about their transmission and how are they really good in the outside barriers, like will they get actually, are they stable or not, these kind of things. So now let's go into chapter two, which was still general. We're going to be talking in generally about just viral replication. And in specific, we want to talk about how just one little virus like this enters one of these massive big cells compared to relative size to them and suddenly becomes hundreds of thousands of viruses, literally. So how are they able to literally photocopy themselves? So we're really going to be talking about that. And when it comes to that, there's two ways of looking at viral replication. There's either you can look at it as a growth curve, or you can look at it in a stepwise manner. And we'll be discussing about the two in specific, but we're definitely going to go much more in details about the stepwise. So when it comes to the growth curve, this is literally how you can summarize how a viral replication looks like. So the y-axis is the amount of viruses per cell, while here is basically the x-axis is the hours. So let's imagine at hour 0, you have one cell being affected. So what happens is you have suddenly this literally like short, low amount. Basically, this virus is at 1 and then it goes to 0, after 1 hour. Then from 1 hour to literally almost 5, there's no virus. There's nothing. This is called the eclipse period. Okay? The eclipse period technically means is that when the virus goes inside, it's of course it's gonna be disintegrated, and then you're gonna have a lot of basically the genomic structures being released, either DNA or RNA, and then it takes time for it to start replicating and creating new virus particles. So technically, by this point, there's no viruses. There's nothing. Only later at basically at the 4 hours, you have nucleic acid, which is seen here by the dotted lines, start to form, and bit by bit, once you have enough nucleic acid, you start to form your viruses. And basically, the eclipse period is done. It's done when you have the first virus coming up. And please don't confuse this period, which we call the eclipse period, with latency. Latency is a bit different, but we will be talking about them a bit later. But latency is something that happens literally when a virus literally goes in, and then basically, the virus also still gets out, but that's the latency period. At this point of the year, the eclipse period is literally is when the virus gets in, and it's there, it's inside. It didn't go out yet. So that's what the difference between eclipse period and latent period. Latent period is already the virus has gotten in and has gotten out, technically. The eclipse, no. It just gotten in and suddenly there's no more viruses because technically now you've disintegrated the whole virus and released the DNA or RNA to replicate. And then later, bit by bit, once you have the nucleic acids start to replicate, virus start to accumulate. And you can actually go from one virus, for example, here in this case, you have 100. But of course, this can be thousands in some cases of viruses. And this is literally in 10 hours. So imagine in 10 hours, you have such a huge amount of virus being produced. And in some cases, in some viruses, what happens is that imagine that you have one cell. You get one virus that comes in and it starts to replicate from one to suddenly hundreds of thousands. Well, these hundreds of thousands will actually kind of disrupt the whole cell morphology. Because think about this, you have a cell, you have a compartment, and it's getting filled and more with a lot of viruses that are literally pushing the cell membrane, and it actually can change the morphology, and it can actually destroy cell functions and the whole actually cell. And we call that cytopathic effect, CPE. Not all viruses demonstrate CPE, by the way. Some of them actually will just come in, replicate, and really bit by bit just butt off the whole cell without really damaging the cell. But there are some ones that will actually do a lot of damage, and that damage is called the cytopathic effect. So this is an example. These are A549 cells. These I know this so well because of this very beautiful kind of rectangular or rhombus-like structures. The A549 cells are lung carcinoma cells. And basically, what they've done here, they've added adenoviruses. So this is literally after, let's say, the next day, you get to see literally this cytopathic effect. So you see now this small, we call these kind of plaques because it's like one little plaque that is literally, you see some cells around that the morphology is getting weird. You see, they're not this really beautiful rhombus shape anymore. No, you actually have this very different kind of morphologies happening over here, and this is what you end up getting. Suddenly, from this very beautiful rhombus shapes to these kind of literally, these cells like look horrible, circular, and quite, let's say, dead per se. So this is actually some of the viruses can cause. So remember, some viruses can cause the CPE effect, cytopathic effect, and some no. Some of them can just not actually do anything with the cell morphology. So now we saw the whole replication of a virus in a curve. So let's look at it in a step manner. So when it comes to viral growth, you have the following literally steps, which literally almost all viruses follow the steps. The first one is the attachment. You have a virus that attaches to the cell of interest, and this is very specific, which we will be talking about. And then the virus penetrates the cell, it goes inside the cell. Once inside the cell, it has to uncoat its viral genome because, as you remember, the viral genome is stuck to the capsid. So you have this formation of a nucleocapsid, which is literally your nucleic acids, which are surrounded by the capsimers. So you need to remove these capsimers away to release that nucleic acid so it's free and then able to do, its replication, translation, and whatever. So then once you have the uncoating and the release of the viral genome, first thing is viral mRNA synthesis. You basically come every single genome, no matter what it is, DNA or RNA, single-stranded, double-stranded, whatever, it's always going to end up with viral mRNA. There's always going to be the mRNA. One, because from the mRNA, you go into your protein synthesis, and then the protein synthesis, here you have all the replication of literally your enzymes, certain things that are going to help you replicate the viral genome, and then they're basically going to help actually, do the viral genome replication. And then you have something called the late viral mRNA synthesis. So you have early, and this early is mostly done for enzymes for the genome replication. And the late one is a bit more later, and this is mostly for the protein synthesis. This is mostly for the capsimers, the ones that are literally creating the new baby virions, so the baby basically kind of viruses. So we'll talk about what this basically early viral protein synthesis is and late protein synthesis. But then basically once you have everything, all the building blocks of the virus, you have literally the capsimers, you have the enzymes, you have what do you call it, the nucleic acids, you create the virus and the virus is released. So we're going to go into a lot of details into every single step. So let's start with attachment. It's the most simplest and most important part because what happens is that, as we said, viruses, be they enveloped or non-enveloped, they literally have these proteins or these glycoproteins that are found on the outside of the virus. And these ones are the ones that are going to be binding to certain receptors in a cell. So what happens is literally is a virus will literally bind to specific receptors. This is specific. So realize here, this is basically here you have, this is your, what do you call it? Influenza virus. It only will bind to sialic acid. It's not going to bind to these other basically structures. No, it's just literally going to bind to sialic acid in specific. So this interaction is specific, meaning that this virus, for example, this influenza virus, is not going to bind to another receptor, for example, a growth receptor. No, it doesn't have that specificity. It's literally, think about it as a receptor and ligand kind of interaction. It's a weak interaction, a non-covalent interaction that kind of signals for the cell to take up this whole virus. So basically, it's this interaction that gives the virus host range, meaning that certain viruses will replicate in humans, some of them will replicate in humans and also dogs, and some of them will replicate in basically everything. It all depends on what the receptors on the virus binds to. So, for example, polioviruses have a certain receptor in them that can only bind in humans and other primates, but that's it. So you never see a dog having polioviruses, you never see a cat having polioviruses because the viruses will not be able to bind to the cell receptors of that dog or cat because there's no interaction with them. But some others, like rabies, rabies doesn't care. Rabies will bind to basically all mammalian cells because it binds to a receptor that is found within all mammalian cells and actually serves a similar homology between all mammalian cells. So this is how this kind of, let's say, receptors or glycoproteins on the basically exterior of a virus can determine the host range. It's extremely important to know that. And also, other than just, of course, host range, it can also govern basically organ specificity. So this is why we have, for example, herpes virus. So now, herpes viruses, for example, they will only bind to certain ganglions. They will not bind to your liver cells or somewhere else because the ganglion cells are only the ones that have the receptor. Or the hepatitis viruses, they will only bind to the liver cells because they're the ones that only have that receptor. The heart doesn't have it, the kidneys don't have it. So this is how these glycoproteins in the virus can determine the organ specificity. So once the virus is able, literally, it's going through your body, it scavenges, it looks to the cell. This is the cell, and basically the virus knows that this is the cell I need to infect because of this receptor from the virus and the cell interaction. And this should basically kind of give a clue either to the virus or mostly to the cell to be like, okay, start to, infect, start to penetrate this cell. And how most of the viruses do it is through something called endocytosis. So endocytosis is literally means invagination of the cell membrane that form vesicles in the cytoplasm. So what literally happens is the virus, in this kind of case over here, is literally gonna bind to the receptor, and that's gonna cause some sort of signaling cascade that kind of invaginates. So invaginates is literally when it kind of you push the membrane a bit down. So that's what invaginates means. So literally like this kind of formation. So it pushes the membrane inside, and it will literally kind of create this kind of vesicle. So you push, push, push until you get this vesicle. Inside the vesicle, this basically vesicle will go through some, changes in pH, which we will talk later. But this is how the viruses usually penetrate, the majority of them. Not all, and this depends on the type of viruses. Okay? So when it comes to enveloped viruses, most of the enveloped viruses actually go through endocytosis, as I said. Others will go through literally something called synthetia. So this is called the examples are this paramayo and the herpes viruses that do that. So what happens is that you have, let's say, the cell that actually got infected by the herpes viruses through endocytosis. But what happens is that the herpes virus will actually start to put these viral proteins over here. So these ones shown in yellow. These ones can fuse to other uninfected cells, and they can fuse all together to create one massive cell that is multinucleated, because it's literally the formation of, literally in this case, 5 different cells together. So suddenly this can affect all the other cells that are now combined together. So this is one way. It's called synthetia. So synthetia literally means multinucleated giant cell. So it's literally a big cell with multiple nucleuses. So that's how one virus can infect multiple other cells, by literally putting their viral proteins in the membrane of the cell and causing this infected cell to bind or fuse with other uninfected cells. For unenveloped viruses, as I said, majority of them go through literally endocytosis, but some can actually form this beautiful pore. Very, very similar to what we see with bacteriophages. And we see this with picornaviruses. Picornaviruses, so in this case here, you have the most important part, the attachment. That attachment kind of signals the virus like, okay, I need to literally kind of inject my whole virus. So it literally has this needle-like structure that pushes through the cell membrane and then threads. It literally threads the whole genome inside to the cytoplasm. So that's how some unenveloped viruses can go. But this is just one example. Majority go again through endocytosis, this invagination. So this is just another example, basically the most important part, as we said. You have the binding. So this is the host plasma membrane, basically, and then you have here the receptors in the host cell plasma membrane. It's gonna bind specifically to these viruses, and that's gonna cause some sort of signaling cascade in the cell that's gonna invaginate this basically kind of cell membrane to pick it up into an endosome. So that's how they're able to penetrate. The majority of them, as we said, some can do syncytia, and the others can cause that basically literally this penetration by this kind of, let's say, pore formation. And then comes uncoating. So once inside these vesicles, you need to make sure that what happens is that the nucleic acids are released, because now in the case of the virus inside here, it's actually part of the of the capsid. You want to kind of remove the nucleocapsid and release just the nucleic acid nakedly, meaning that it is not covered in anything. So let's take this example here with influenza. Influenza will literally bind, it's going to cause endocytosis, and then you're going to get the formation of the endosome. And this endosome, what happens is that certain other vesicles are going to come here. They're going to actually cause this whole this kind of proton influx, this specifically hydrogen and potassium influx inside, and especially proton here is going to cause this to acidify. So suddenly you have from a pH of 6.5 to 6 down to 5. And this low pH kind of causes some sort of, let's say, change in the virus. And the virus is going to note that change, and either what's going to happen is that the virus is literally going to rupture the whole basically pH ruptures this kind of vesicle and ruptures the virus inside of it, and it's going to release the naked protein outside, the naked nucleic acid, or it's going to cause basically certain proteins in the virus to kind of literally this kind of destroy the whole endosome and release the nucleic acid basically. So it's literally the low pH that causes this uncoating. So that's what you have to remember. Not really specific the examples, but I want you to understand that it's once inside the endosome, it's the pH that is causing the kind of uncoating by rupturing technically this whole virus and the vesicle to release your nucleic acids. Of course, in the case of the synthetia, you don't need that because it's already in the cytoplasm. And in the case of basically here, this kind of pore formation, it's already in the cytoplasm, so you don't need it. But this is only mostly happening for viruses that go through endocytosis. So one thing that I want to also highlight here is, there was a certain time that we didn't know in viruses what was let's say the genetic material or this material that was hereditary. We didn't know, was it the protein or was it the nucleic acids, and there was a lot of experimentation to really kind of decipher it. But the most important experiment that really led for us to say that, that whatever is actually you know being in the hereditary kind of molecules is actually DNA. It comes from the Hershey-Chase experiments. So what Hershey and Chase did were two scientists. They got two bacteriophages. One bacteriophage they basically labeled with sulfur, and the other one they labeled with phosphorus. So sulfur is highly found in proteins. So it basically kind of, as you can see here, that's labeled in red. The sulfur basically will only be found in your protein. It's not in your DNA. While in the case of phosphorus, if you label it green, it's only going to be in your DNA because proteins don't have phosphorus, it's only DNA. So they wanted to see what is the thing that is going inside the cell. So it would be super easy for them because what they would do is they would take the cells, first of all, put the bacteriophages, let them infect, and then see inside the cell what is there. Is there something red or is there something green? So this is literally what they did. They first got the infection, they let the infection occur, and then they blended. And what the blending did is they separated literally the viruses from the cells, and then they centrifuged. And when you centrifuge, basically you spin something at a certain speed, what you can do is you can push heavy stuff down. Because viruses, these bacteriophages, are very, let's say, negligible in weight compared to the cells. The cells will literally sediment or literally will go into the or sediment into the bottom of a tube. So then basically you can remove the supernatant that contains the viruses, so you can remove it, and inside that basically you have the sediment, you can check. And they realized that in the case where you actually label sulfur, there was no red, there's nothing. But in the case of the green, there was actually inside the cell. So this kind of told them like, aha, what is actually the infectious or the literally the thing that is hereditary that's entering the virus, sorry, the cell, and replicating is DNA. And it's literally DNA that is the thing that is actually the thing that is literally, the hereditary element that a virus possesses. And why I brought this whole experiment back is to actually introduce a very nice thing that's called infectious nucleic acids. So here, in this kind of sense, we're talking about a whole virus that has to bind to the cell, enter through endocytosis, and basically literally rupture to release its DNA or RNA. Well, some basically viruses can actually not use the virus per se. They've realized that sometimes when you purify genomes, they can be infectious on their own. So they saw that in certain cases, some DNA or RNA, you can actually carry out the whole entire viral growth without the protein. Meaning that, for example, if I take literally, in the case of, let's say, polio, if I take polio's, just its nucleic acid, I can literally put this in a cell. This basically nucleic acids will be able to penetrate into the cell and actually cause infection without the need of the whole virus. So they call this infectious nucleic acids. So nucleic acids that are infectious, that on them by themselves, they can penetrate and start creating new viruses or infect. But remember, all viruses are infectious, but not all DNA or RNA are infectious nucleic acid. So remember that. So all viruses are able to basically bind and penetrate the cells, but not all DNA and RNA is able to penetrate the cells and actually replicate. So this is one thing you have to remember. And we will be talking about specifically why not all of them, because there's a very particular reason, and that really involves on how the certain machineries that is needed or not. But why is this interesting? Why is infectious nucleic acids an interesting point? Well, it's quite interesting because the observation that purified nucleic acid is infectious is the definitive proof that nucleic acid, not protein, is the genetic material. This really kind of nailed the freaking nail to the coffin, to the whole dilemma. What is the thing that is causing and basically hereditary, or let's say, that is infectious? Is it really the protein or is it the DNA? No, it is the nucleic acid that is the genetic material. That's the one that is basically the hereditary element. Second, which is super cool actually and dangerous, is that infectious nucleic acids can bypass the host range specificity provided by the viral protein cell receptor interaction. So remember, viruses are governed by this viral protein cell receptor interaction. So as we said, in the case of polios can only infect in certain primate cells or humans. But it cannot, for example, grow in basically in a cat cell or a dog cell. But if you take the RNA from the poliovirus, it can actually enter non-primate cells and go through usual growth cycle. So technically, the poliovirus cannot actually infect a canine cell, but the infectious RNA of polio can do so. So it can actually bypass this whole host range specificity that's governed by the viral protein receptor interaction. This is quite interesting. Third one, only certain viruses yield infectious nucleic acid. The reason for this is discussed later. So remember, as I said, not all viral DNAs or RNAs are infectious. So we will be discussing why in that point a bit later. But then You finally have your nucleic acids that are in your cell. Is they released? Every single virus, no matter the type of nucleic acid, be it DNA or RNA, double stranded, single stranded, positive polarity, negative polarity, no matter what, they're all going to basically go into viral mRNA. The way that they go there, it obviously depends on the nature. If it's double stranded, it's DNA or RNA. If it's basically positive or negative, basically strand or polarity. And we will be talking about how every different virus is able to go into viral mRNA. So in the case of DNA viruses, basically with exception of one DNA virus, they all replicate in the nucleus, because what happens is that basically the virus goes inside, it will go to the nucleus, because inside the nucleus you have your DNA dependent RNA polymerase. So they're able to go into the nucleus to create your mRNA, because that's where the machinery is found. However, as I said, there's one exception, and that's the pox virus. The pox virus is not able to carry basically the replication inside the nucleus, because this one is quite weird in a sense. So this is basically what happens: it binds, it releases its DNA, and it actually has on its own, it has basically its own polymerase. It has its own polymerase that it's able, so shown basically here, this kind of, let's say, little blue kind of dot, that's able to actually induce mRNA production by themselves. And one thing that is quite interesting also to know is that, remember, all DNA viruses are generally double stranded. There's only one exception, which is the parvoviruses. Those are single strand DNA. But all DNA viruses are usually double stranded DNA. And those usually are quite simple. Basically, they all super easily can go into mRNA by using the cell machinery's uh, DNA dependent RNA polymerase. It's only the exception of this kind of pox virus that it likes to go in the, let's say, cytoplasm. RNA viruses are different in a sense that they actually love to replicate in the cytoplasm, because they don't need to go to the nucleus, because they don't need the DNA polymerases. They don't need this DNA dependent RNA polymerases. They're not really in need of that. So most RNA viruses, as I said, they do it in the cytoplasm. There are exceptions like the retroviruses and influenza viruses that do not. They will basically like to, and basically are mostly replicated in the nucleus. And I'll be explaining a bit more why those two are a bit particular. But all the genome of RNA viruses usually is consisting of single RNA stranded. There's only one member, one, let's say, virus family that is double stranded RNA, which is the reoviruses. These are the ones that cause rotavirus, that cause this rota infection, that is extremely annoying, because it's the one that causes you to vomit and let you have diarrhea at the same time. It's quite annoying. But these are RNA viruses. But I want to give you basically why retroviruses and influenza viruses like to replicate in the nucleus. So as we know, retroviruses, when they enter, they basically have a RNA, basically an RNA genome. It goes in basically to the nucleus because it has its own, literally, a reverse transcriptase that is able to basically release DNA that is basically needs to go inside the chromosome, basically the cell's DNA, so it integrates. So that's why it replicates inside the nucleus, because it needs to integrate its DNA inside the cell's DNA. Influenza viruses is a bit different. Basically, once they infect, they release their mRNA, literally segmented RNA kind of pieces. So remember, the case of influenza, they have actually their RNA is into pieces. It's not one big, what do you call it, circular kind of piece. It's no, it's literally multiple pieces. So the mRNA, it likes to go inside there because the influenza has its own RNA dependent polymerase. So remember, when it comes to humans, we do not have RNA dependent RNA polymerases. We have DNA dependent, which that means is that we need to have DNA basically as a, and generally as a strand to create mRNA. We cannot create mRNA from mRNA. But the influenza virus has actually basically an enzyme that's able to do that. And it goes inside to the nucleus where it actually starts to create the mRNA synthesis for protein translation, but also for replication. So, in generally, RNA viruses fall into four different groups. Basically, you have the single RNA positive polarity, the single RNA negative polarity, double RNA, and single RNA of positive polarity that is transcribed into double-stranded DNA. And these are the retroviruses that we talk about. But I'm going to go into details into each one and how they basically will produce their mRNA. So when it comes to single-stranded RNA with a positive polarity, these are extremely easy. Literally, their basically genome already serves directly as mRNA because it already is a positive sense, so it already can basically be used to basically translate. So that's the beauty of single-stranded RNAs. When it comes to single-stranded RNA with a negative polarity, no, they need their own RNA-dependent RNA polymerases because, as I said before, humans do not have RNA polymerases that can actually transcribe mRNA from RNA. It can do it from DNA, so hence it's DNA dependent. But in the case of single-stranded RNA with a negative polarity, the virus has to have its own. They need it, which is this RNA-dependent RNA polymerase. So that's able to kind of transcribe. So you have this is your viral RNA, so hence the V is there for RNA, the negative strand. Then basically the RNA-dependent RNA polymerase is going to then transcribe it into an mRNA with a positive. So that's how they reach to this mRNA. You also have certain hybrids. These are really weird, certain, how can I put it, viruses in a way that the region consists of regions with a positive polarity and regions with a negative polarity. So as you can see literally here, these are literally this kind of bunia viralis, articuloviralis, serpento. These are called ambisans, and they're very weird, honestly. These basically still need their own polymerases because you still need, in some sort of way, um, the RNA dependent polymerases, but they have basically both positive and negative polarities. But then you have your double stranded RNAs. These basically, double stranded RNAs still require the virus to carry its own polymerase because despite the double stranded RNA has the positive polarity, which can then be used to do transcribe, it's stuck by hydrogen bonding to another RNA. So that per se is not be you cannot actually be able to use it. You need actually another polymerase that can actually take that double stranded and actually transcribe a positive basically polarity of an mRNA. An example for this usually is rotaviruses that does that a lot. And the final one is literally the single-stranded RNA that converts into DNA. So these guys, they take their RNA and they basically convert it into DNA, and they use the reverse transcriptase. And then from DNA is then able to basically transcribe into viral mRNA by the host cell's RNA polymerase. Because again, now basically we have this single cell RNA that's now converted to DNA, and we know that in the host cell, specifically in human cells, we have DNA dependent RNA polymerases. So we can convert this DNA into mRNA. So great. But of course, the viruses need reverse transcriptase. We don't have these kind of enzymes with us. And these big two that actually use this literally retro kind of transcriptases, basically, or reverse transcriptase, are generally the retroviruses and the hepadna. So retroviruses are like HIV, and the hepadna in specific is hepatitis B viruses. But they use it in a very different sense. As we said with retroviruses, we know that basically they like to, transform the RNA into DNA really early on to replicate their genomes. So this is really, really early on. Hepadna, they do this really late. And hepadna, specifically the hepatitis B virus, it's quite weird. So let me show you how the infection of hepatitis B really works. It's quite interesting. So here you have the hepatitis B virus. It then basically attaches. It does basically the invagination, the cytosis, the release of your basically the viral DNA, which in this case is literally these kind of DNA chunks over here, which in some way are actually kind of uh, in some way, how can I put this? Kind of have missing sections. They're actually broken. They're not fully really well adjusted, or let's say robust. So they're quite broken. So what happens is that they actually have a, let's say, what's it called, a RNA polymerase that is able then to basically, first of all, or DNA polymerase that's able to actually turn this or circularize this and actually fix the whole missing sections. Then this DNA is able then to be turned into RNA by the host machinery. But then the mRNA is then basically reverse transcribed into DNA to basically, to replicate its genome, because the genome here basically is DNA. So notice here that the DNA dependent DNA polymerase that basically exists within the what's it called, HPV, has both DNA dependent and RNA dependent activities. So DNA dependent activity is at the beginning. Remember here when we had the DNA, it's able to take this and literally synthesize a new piece of DNA that is now basically circularized and fixed because this was broken. And here you take the RNA, and then you basically, it's now RNA dependent because now it's turning this RNA into DNA. So it has these two very beautiful kind of properties: DNA dependent and RNA dependent DNA polymerases. So this is basically HPV. This falls within this category, but it's different to the retroviruses in that kind of sense. Retroviruses really use more the RNA dependent kind of activity, while the hepadna uses the double. And basically the retroviruses, usually this happens really at the early replication cycle. This happens way later, basically. So let's go back again to remember when I was saying that only certain viruses yield infectious nucleic acid. The reason, as I said, is going to be discussed later. Well, the reason for this is simple, is that viruses that do not require a polymerase in the virion can produce infectious DNA or RNA. So remember that some viruses do not need extra proteins or polymerases to replicate their genome because they use the host cell's proteins to do that. But some, like herpes, the hepadna, the other ones that I was talking about, they require their own RNA, basically RNA dependent RNA polymerases, basically, or these other ones that the host cell doesn't actually have. So if you throw in those basically RNAs or DNAs, they're not really going to replicate because the host cell doesn't have the proteins for to actually replicate that. So let's say in the case of DNA viruses, so we know that the single-stranded DNA viruses basically can actually be infected, have an infectivity, like can have, let's see, can be infectious nucleic acids because the virus, basically the cell host, actually has RNA polymerases that can bind to the DNA and cause it actually to what's it called, replicate. In the case, for example, as the circular partially single-stranded, in the case of hepadna, no, we cannot, because remember that hepadna, the hepatitis B viruses, they need to convert the mRNA to DNA, but we don't have RNA-dependent DNA polymerases. That doesn't exist with us. So that's why it doesn't have basically infectious nucleic acid. Same thing goes if you look at the RNA genomes. So if you look at the single-stranded, with a positive polarity, these can be infectious genomes because we have literally RNA-dependent RNA polymerases, basically. Sorry, we have basically, that can basically cause this to, and basically infect. But in the case of, these kind of RNA-stranded, segmented or segmented, basically with the negative polarities, they're not infective, not at all, because we don't have literally the enzymes to replicate them, because we don't have an RNA dependent polymerase. We have a DNA dependent. So once we have the mRNA, that's out and done, now comes translation. And translation, as I said before, you have two basically ways. You have the early proteins and the late proteins that are done. So what defines early proteins or late proteins? It's literally, we define literally the replication of the genome in the middle. So anything that happens before the replication of the genome is called early, and anything that happens after is we call that literally, late. And in generally, the early proteins that are produced at the beginning are enzymes that are required for replication of the viral genome. These are the ones that are really needed, the polymerases and all that kind of stuff that help us, like, create a lot of these different types of basically to replicate the genome. Then later, the late proteins are more the structural proteins. These are the nucleocapsids, sorry, the capsimers, and these things that are going to be creating your whole virus. And when it comes to protein production, some viruses, basically, some will create mRNAs that are translated directly into structural proteins. So basically, you create one mRNA that gives you one protein. Some are quite weird that will actually translate into a whole precursor polypeptide. So literally one viral mRNA is going to give you one massive precursor polypeptide that is then cut into smaller proteins, and these are different proteins with different functions. So I'll give you examples for that. So this is basically for HIV and the picornaviruses, but this is as a nicer picture. So in the case of the polioviruses, for example, here you have the translation and you have this one massive protein that's literally like that. So what happens is that you actually get these virally encoded proteases that come in and they literally cut the proteins and give you from one big massive precursor protein into three different proteins with three different functions. Retroviruses also have the same kind of thing that they can actually give you in this kind of case here a whole precursor protein like the GAG and POL. So these are really needed for polymerase for replication. So they basically you get one big precursor protein that is then cut, and you have the GAG protein out and the polymerase protein out. So either that you have one mRNA creates one protein, or you have one mRNA that creates one big massive precursor protein that is cut into pieces that give function to many different proteins. And then after basically that, you have the replication of your viral nucleic acid. So that's the most important thing, because if viruses want to still be infectious, they need to replicate their genomes, because every newly formed virus needs to have a genome. And the genome is basically governed by a principle of complementarity, meaning that once you start with the genome, basically the parental genome, for example, and let's take the example of poliovirus, the parental genome is single-stranded RNA. So this single-stranded RNA, basically, we need to duplicate this. So the progeny genome has to be positive single-stranded RNA. But we cannot just duplicate it literally by single-stranded RNA. Just like, taking basically a polymerase and just copying it. No. We need to form an intermediate form of it. So we need to actually have a polymerase that comes in and gives you this intermediate form, this negative single-stranded RNA. And then this is going to be used then to copy and create the positive single-stranded RNA. So this is by what I mean by complementary principles. So that's in the case of polioviruses. In the case of influenza, where you have a negative stranded single RNA virus, you need to create the positive single stranded RNA, that which then gives literally, again, your single stranded RNA. Double stranded RNA, basically, RNAs, basically in this case, you can actually create a positive stranded single stranded RNA, and then you create again the double stranded. Retro, positive single stranded RNA. In the case here, their intermediate form actually is weird, because as we know, they actually go to reverse transcriptase, they go to DNA, and then the DNA can be used as the intermediate form to create your single-stranded RNA again by complementarity. The paroviruses, these that have the single-stranded DNA, can basically go, copy it basically into a double-stranded DNA, and then from there you go into single-stranded RNA. The hepatitis B viruses can use its basically mRNA, its double-stranded DNA to create RNA, as I remember, to the intermediate form, and that then is basically then turned into again to double-stranded DNA, because this we basically spoke a lot about. The papoviruses is double-stranded DNA, which basically you can actually use again, create a copy it by the host cell machinery, and again you can copy it again at the same time with the progeny genome as well. And then finally, you have the assembly. So the assembly is literally now when you have all the proteins, you have all the genome that's replicated, and you basically they all come together, they assemble into a new virus, a virus that has now a nucleocapsid that is basically inside, the basically nucleic acids and everything. The precise steps actually of how this has happened is very little known. We don't know much about it. But what we know is that if you take these RNA or DNA, so basically if you purify the nucleic acids and you purify the viral proteins and you put it together in a test tube, they'll assemble together without the need of enzymes or any energy. So it's quite, let's say, quite cool in a sense that they don't need much energy or, let's say, kind of help to do this. And then the last thing is release. So basically, the release happens by two ways. One is by rupture of the cell. So this is the cytopathic effect, in where you literally have these cells literally multiplying in so much huge numbers, and they're just literally destroying the whole cell, and literally they're removing themselves by force. They're rupturing the cell membrane, which is then basically kind of vomiting out the new formed viruses. The other one is called budding. And budding, what happens in general is you have the glycoproteins of the virus. They go into the membrane of the cell, and then basically the matrix protein comes in and actually interacts with these glycoproteins, and then you have the basically nucleocapsid. That the nucleocapsid is then able to actually interact with these basically glycoproteins of the viruses by the formation of basically or by the M protein. So you basically this is kind of pushing. So you have basically this imagination again of your vesicle, and then you basically are pushing it, and that's how these viruses are stealing the whole outer cell membrane. So this is the whole process of budding. In the case of herpes viruses, they don't actually do the budding through the cell membrane; they do it through the nuclear membrane because they basically are replicating in the nucleus. So now the final thing I want to talk about is lysogeny. This is a bit of a different pathway. It's an alternative pathway. It's not this the pathway that we've seen that these basically viruses they come in, they attach, they release their viruses, sorry, their nucleic acids, they start to translate, they have your mRNA, you have now suddenly your basically proteins, and you suddenly get new virus particles, which then, either rupture the cell or they bud off. These ones actually is that the viral DNA actually becomes integrated into the whole um, host DNA. And a good example of this is the bacteriophage, this kind of lambda bacteriophage. So just like other viruses, they can actually come in and infect, replicate, and literally kind of destroy this whole damn cell. But in some cases, in the lysogenic cycle, it can actually integrate its whole DNA into the host genome, and they can basically lay there dormant without producing not a single particle for days, months, or years, whatever you want. And this is quite interesting because the medical implications of this is how certain bacteria develop exotoxins. So these toxins like diphtheria, botulism, cholera, these toxins that when bacteria release can be extremely deadly to us, you know? So like in the case of botulism, it can actually really cause a lot of paralysis in us. So this is how certain bacteria that don't have genes of botulism acquire it. So here's an example. You have, for example, here this kind of phage, which has its own phage gene, but it also has basically the diphtheria toxic gene in it as well. So what can happen is that this basically bacteriophage will release its DNA, and then this DNA will integrate inside of basically this bacteria's whole genome. And now this bacteria has the diphtheria toxin. So now it acquired a new kind of function to create actually diphtheria toxins. And that's how certain bacteria can actually acquire these very pathogenic um, characteristics through these phages, actually. And this whole thing is literally called lysogenic conversion. So, lysogenic conversion, it means that literally you have this bacteria that gains new properties because of the integration of a phage gene that basically here over here. So you basically have the whole phage gene integrating into the host genome. And basically now the phage genome that is integrated in the host genome, that's called prophage. So the genes that are literally integrated into the bacteria, basically phage. So bacteria, sorry, the prophage literally means it's the DNA that is basically kind of now incorporated into the host genome. And this is also called, other than lysogenic conversion, it's called transduction. It means that there's a transfer of genes from one bacteria to the other through viruses. So we do this actually quite a lot in the lab. Sometimes we want to actually give a virus a new gene. So what we do is we use bacteriophages or we use other viruses to do this basically, to actually transfer, to give our genes into one. So it's called transductions. But basically, you have this lysogeny or lytic kind of cycles. What determines that when a basically bacteriophage infects, what determines if it's going to go into the lytic cycle or if it's going to go to the lysogenic cycle? Well, it all depends on what two proteins are being produced. Is the repressor protein being produced or the antagonizer? So in these bacteriophages, you have repressor proteins, which are called CI genes, and you have antagonizer repressor proteins, which are called CRO genes. If, in the case of the bacteriophage, you have a lot of repressor genes being produced, this repressor genes is going to stop any actually of protein synthesis. So it literally binds to operator sites in its whole genome and it's going to stop any proteins from happening. So it kind of pushes it into the lysogeny phase. If the CRR or the CRO gene is being produced quite highly and much more than the CI gene, the CRO is able to bind to the CI and kind of inhibit it. So then protein translation is actually starts to happen, and then you enter the lytic cycle. So this is literally, the whole, how can I put it, kind of process of how this happens actually. So the whole lysogeny phase happens. So what happens is that you get actually these bacteriophages that literally attach to the bacteria through certain receptors. They inject through pores their DNA, and their DNA is usually actually linearized. And what happens is that then basically inside is actually circularized. So they kind of make it into a circle. Then, because once it's into a circle, you have certain enzymes that's going to break your host genome. So seen over here, it's going to break the host genome and it's going to basically kind of integrate this circular DNA inside here. So remember that DNA has to be circular for it to actually go to kind of join. If it's linear, it doesn't join. So that's why it has to be circularized. So once it actually joins, you have now the prophage. So remember, the prophage is the DNA of the bacteriophage that's integrated with the host DNA. And then basically this can stay here for days, weeks, years, you name it. But there's sometimes certain things that can push into the lytic cycle, and that's UV radiation. Because UV radiation kind of stops these repressor genes that I was talking about and kind of allows protein translation and then to go into the lytic phase. And once you get to the lytic phase, you get the production of basically bacteriophages. But what happens technically is that this prophage is then excised, removed, and then basically replicated and again be taken and put into the newly formed basically bacteriophages. So sometimes what happens is that the prophage is excised correctly and you get these very infectious bacteriophages, but sometimes it's excised incorrectly in that in this case here you would take a bit of the bacteria DNA and it's encoded basically in this bacteriophage, and this bacteriophage suddenly is not infectious because it doesn't have its full genome. It has now a bit of the genome of the E. coli here, so it's not infectious. So as I said, UV radiation is actually something that can cause uh, the whole lysogenic phase to go into lytic phage. It kind of pushes the prophage to be removed and to start to translate and create new viral or more bacteriophages. So when it comes to lysogeny in bacteria and latency in human cells, so we really know that lysogeny happens in bacteria quite often, but when it comes to the latency in human cells, this is basically where you have viruses in human cells that they actually will infect, will integrate into the genome, and they will stay there. So very similar to lysogeny. So you have a very clear parallel happening there. With lysogeny, we know what's happening really well, but with latency in the human cells, it's not really well known. So most of these latency viruses are most of the ones coming from the herpes virus family, like the herpes simplex virus or the cytomegalovirus or the Varicella zoster, the Epstein-Barr viruses. These ones exhibit latency. And as I said, you have very to little no virus produced at all. And in the case of HSV, we know that it has this production of these kind of transcripts, these mRNAs, basically that are called latency associated transcripts. And what they do is they really kind of suppress viral replication. How it does that, we don't know. We really don't know what happens, honestly. And we don't know how these things are basically being produced and how they're being controlled. It's quite a mystery. Something that a lot of people in the let's say herpes simplex virus kind of domain are trying to investigate a lot. The cytomegalovirus employs a different thing. It actually has these microRNAs, and microRNAs, what they do is they inhibit translation completely. These are transcripts that kind of suppress viral replication. This actually suppresses the translation of mRNA. So microRNAs are really well known to kind of push the host cell machinery to cut mRNAs and not allow them to replicate or translate into proteins. So this is basically the end of chapter 2.